Besides column chromatography for large-scale isolation of plasmid DNA, there exist other methods which can give you good yield of the DNA. Cesium chloride density gradient is one of such method. A density gradient of cesium chloride is achieved by centrifuging the cesium chloride solution at very high speed. And if any macromolecule mixture is present in that tube, the macromolecules will settle as per their density in the tube or in the gradient of cesium chloride. Different molecules, they have their different densities, which is referred as buoyant density. The DNA, the double-stranded DNA, has a buoyant density of 1.7 gram per centimeter cube, which helps the DNA to settle at a particular point in the gradient. On the other hand, protein has less density. It will float on top of the gradient, whereas RNA will settle at the bottom. This figure shows you that how we can establish a density gradient of cesium chloride by taking the solution of cesium chloride in the tube and then doing the centrifugation at higher speed. When we load the sample containing the macromolecules including protein, DNA and RNA, and again we centrifuge it, DNA settles at its respective density position, which is 1.7. Protein being having the less density, they are floating on top of the gradient, whereas RNA settles at the bottom. This method has benefits over the column chromatography method. This can be used as an alternative in order to avoid the use of organic solutions which are involved in column chromatographic methods. This method is very useful to get highly purified DNA with high concentration. Besides getting the high concentration, this method is also useful to separate the supercoiled DNA from non-supercoiled DNA. As you know that ethidium bromide intercalates in the double-stranded DNA, so if we use ethidium bromide, the supercoiled DNA gets ethidium bromide incorporated in it, and if this is a circular molecule, there is limitations in unwinding of supercoiling or supercoiled DNA. So this is possible that using this method we can separate the linear molecules or the relaxed molecules from the supercoiled molecules. Because intercalation of ethidium bromide leads to decrease in buoyant density, and that decrease in buoyant density will make it possible that the two types of DNA, they settle at different positions. And this difference in position is because of unwinding of the DNA due to incorporation of ethidium bromide. This picture shows you the separation of linear or open circle and supercoiled DNA, and this is because of treatment with ethidium bromide. Once we do this, there is possibility to separate and to purify the supercoiled DNA. Once we are successful in separating the supercoiled DNA, then we have to proceed further so that we can remove the cesium chloride from it and we can get rid of ethidium bromide from our DNA sample. To remove the ethidium bromide, we have to use an organic solution or organic solvent, that is N-butanol. N-butanol can remove the ethidium bromide. In the later step, the dialysis bag is used to get rid of the cesium chloride in the presence of a buffer. The supercoiled, non-supercoiled DNA bands can be visualized under UV light, and the shining band appears, and this is because of intercalation of ethidium bromide into double-stranded DNA. The pure plasmid can be removed by puncturing the gradient tube. It is separated. Subsequently, before we have to use it for further experiments, we can get rid of, and we have to get rid of ethidium bromide. We have to remove cesium chloride and get our DNA in purified form, which can be used for subsequent experiments.